Chapter 3 Plant Kingdom. In the previous chapter, we look at the broad classification of living organisms under the system proposed by Whitaker in 1969, wherein he suggests the five kingdom classification via Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classifications within Kingdom Plantae, popularly known as Plant Kingdom. We must stress here that our understanding of plant kingdom has changed over time. Fungi and members of Monera and Protista having cell walls have now been excluded from Plantae, though earlier classification placed them in the same kingdom. So the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperm and angiosperms under Plantae. Let us look at classification within angiosperm to understand some of the concerns that influence the classification systems. The earliest classification of is used only the gross superficial morphological characters such as habit, color, number and shape of leaves, etc. They were based mainly on vegetative characters or on androecium structure system given by Linnaeus. Such systems were artificial. They separate the closely related species since they were based on a few characteristics. Also, the artificial systems give equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against this, natural classification system developed which were based on natural affinities among the organism and consider not only the external features but also the internal features like ultrastructure, anatomy, embryology, and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on evolutionary relationships between the various organisms are acceptable. This assumes that organisms belonging to same taxa have a common ancestor. We now use information from many other sources too to help resolve difficulties in classification. This became more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the character and the data are then processed. In this way, each character is given equal importance, and at the same time, hundreds of characters can be considered. Cytotaxonomy that is based on cytological information like chromosome number, structure, behavior, and chemotaxonomy that is based on chemical constituent of the plant to resolve confusions are also used by taxonomies these days. 3.1 Algae. Algae are chlorophyll bearing, simple, thalloid, heterotrophic, and largely aquatic, both fresh and marine water organisms. They occur in the variety of other habitats, moist stones, soil, and wood. Some of them also occur in association with fungi, lichen, and animals, example on sloth bear. The form and size of algae is highly variable, ranging from colonial form like holwocks and filamentous form like eulothrix and spirogyra. Figure 3.1. A few of marine forms such as kelp as form massive plant bodies. The algae reproduce by vegetative, asexual, and sexual method. Vegetative reproduction is by fragmentation. Each fragment develop into a thallus. Asexual reproduction is by the production of different types of spores. The most common being the zoospores. They are flagellated, motile, and on germination give rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix or non-flagellated, non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Eudorina is termed as anisogamous. Fusion between one large non-motile static female gamete with a smaller motile male gamete is termed as oogamous. Example volvox and fucus. Algae are useful to man in a variety of ways. At least a half of total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy-rich compounds, which form the basis of food cycles of all aquatic animals. Many species of Porphyra, Laminaria, and Sargassum are among the 17 species of marine algae used as food. Certain marine brown and red algae produce large amounts of hydrocolloids, water-holding substances. Example, algin, brown algae, and caragen, red algae, which are used commercially. Agar, one of the commercial products obtained from Geladium and Gracilaria, are used to grow microbes and in preparation of ice creams and jellies. Chlorella, a unicellular algae rich in proteins, is used as food supplement even by space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae, and Rhodophyceae. Chlorophyceae. The member of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass green due to the dominance of pigment chlorophyll A and B. The pigment are located in a definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoid located in the chloroplast. Pyrenoid contain proteins besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made up of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospores produced in zoosporangia. The sexual reproduction shows considerable variation in the types and formation of sex cells, and it may be isogamous, anisogamous, and oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Eulothrix, Spirogyra, and Zara. 3.1.2 Phaeophyceae. The member of Phaeophyceae or brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branch filamentous form ectocarpus to profusely branched form as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll AC, carotenoid, and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment fucoxanthin present in them. Food is stored as complex carbohydrate, which may be in the form of laminarin and mannitol. The vegetative cell have a cellulosic cell wall, usually covered on the outside by a gelatinous coating of algin. The protoplast contain, in addition to plastid, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a strip, stalk. The strip and the leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in the most brown algae is by biflagellated zoospores that are pear-shaped and have two unequally laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, and oogamous. Union of gametes may take place in water or within the oogonium species. The gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common form are Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. Figure 3.1b. 3.1.3 Rhodophyceae. The member of Rhodophyceae are commonly called as red algae because of the predominance of the red pigment are phycoerythrin in their body. Majority of red algae are motile with greater concentration found in warmer areas. They occur in both well-lighted region close to the surface of water and also at great depth in the ocean where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as fluorine starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spore and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization developments. The common member are Polysyphonia, Porphyra, Gracilaria, and Geladium. Let us see the table. Table 3.1 Division of algae and their main characteristic. Classes. It is subdivided into three classes, that is Chlorophyceae, Phaeophyceae, and Rhodophyceae. The common name of Chlorophyceae is green algae, Phaeophyceae is brown algae, and Rhodophyceae is red algae. The major pigment present in Chlorophyceae is chlorophyll AB, in Phaeophyceae chlorophyll AC and fucoxanthin, in Rhodophyceae chlorophyll AD and phycoerythrin. The food stored in Chlorophyceae is starch. The food stored in Phaeophyceae is mannitol and laminarin. Food stored in Rhodophyceae is fluorine starch. The cell wall composition in Chlorophyceae is cellulose. In Phaeophyceae is cellulose and algin. In Rhodophyceae, cellulose, pectin, and polysulfate ester. Flagellar number and position of insertion. In Chlorophyceae, 2 to 8 equal and apical. In Phaeophyceae, 2 unequal and lateral. In Rhodophyceae, flagella are absent. Habitat. In Chlorophyceae, freshwater, brackish water, and salt water. In Phaeophyceae, freshwater, rare, brackish water, and salt water. In Rhodophyceae, freshwater, some, brackish water, and salt water most. 3.2 Bryophytes. Bryophytes include the various mosses and liverworts that are found commonly growing in moist shaded areas in the hills. Bryophytes are also called amphibia of plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in plant succession on bare rock soil. The plant body of bryophyte is more differentiated than that of alga. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoid. They lack true root, stem, and leaves. They may be possess root-like, leaf-like, and stem-like structure. The main plant body of bryophyte is haploid. It produces gamete, hence is called a gametophyte. The sex organ in bryophyte are multicellular. The male sex organ is called antheridium. They produce biflagellated anthrazoids. The female sex organ called archegonium is flask-shaped and produce a single egg. The anthrazoid are released into water where they come in contact with archegonium and anthrazoid fuses with the egg to produce a zygote. Zygote do not undergo reduction division immediately. They produce a multicellular body called a sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nutrition from it. Some cells of sporophyte undergo reduction division, that is meiosis, to produce haploid spores. These spores germinate to produce gametophyte. Bryophyte in general are the little economic importance, but some mosses provide food for herbaceous mammals, birds and other animals. Species of sphagnum, a moss, provide peat that have been long been used as fuel and as packaging material for transshipment of living material because of their capacity to hold water. Mosses, along with lichens, are the first organism to colonize rocks and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plant. Since mosses form dense mat on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. 3.2.1 Liverworts. The liverworts grow usually in moist, shady habitat such as banks of streams, marshy grounds, damp soil, bark of trees, and deep in the woods. The plant body of a liverwort is thalloid. Example, Markantia. The thallus is dorsiventral and closely appressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structure. Asexual reproduction in liverwort takes place by the fragmentation of thalli or by the formation of specialized structure called gymi, singular, gyma. Gymi are green, multicellular, asexual bud which develop in small receptacles called the gymi cup located on the thalli. The gymi become detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organ are produced either on same or different thalli. The sporophyte is differentiated into a foot, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophyte. 3.2.2 Mosses. The predominant stage in the life cycle of moss is the gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from a spore. It is a creeping green branch and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright cylinder axis bearing a spirally arranged leaves. They are attached to a soil through multicellular and branch rhizoid. This stage bears the sex organ. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organ antheridia and archegonia are produced at the apex of the leafy shoot. After fertilization, the zygote develop into sporophyte consisting of a foot, seta, and capsule. The sporophyte in moss is more elaborate than that of the liverwort. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum. Look at the figure 3.2. 3.3 Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purpose and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plant to possess vascular tissues like xylem and phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophyte, the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophyte, the main plant body is a sporophyte which is differentiated into true root, stem and leaves. These organs possess well differentiated vascular tissue. The leaves in pteridophyte are small macrophyles as in Selaginella and large as macrophyle as in fern. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophyll. In some cases, sporophyll may form distinct compact structure called strobili or cone, Selaginella and equisetum. The sporangia produce spore by meiosis in spore mother cell. The spores germinate to give rise to inconspicuous, small but multicellular small but multicellular, free-living, mostly photosynthetic thalloid gametophyte called the prothallus. The gametophytes requires cool, damp, shady place to grow. Because of the specific restricted requirement and the need for water for fertilization, the spread of living pteridophyte is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organ called antheridia and archegonia respectively. Water is required for the transfer of anthrazoids, the male gamete released from the antheridia to the mouth of archegonium. Fusion of male gamete with the egg present in archegonium result in the formation of zygote. Zygote thereafter produce a multicellular, well-developed sporophyte, which is the dominant phase of the pteridophytes. In majority of pteridophytes, all the spores are of similar kind. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two different kinds of spores, macro large and micro small spores, are known as heterosporous. The megaspore or microspore germinate and give rise to female and male gamete to fight respectively. The female gametophyte in this plant are retained on the parent sporophyte for variable periods. The development of zygote into young embryos takes place within the female gametophytes. This event is a precursor of seed habit considered as an important step in evolution. The pteridophytes are further classified into four classes: Silopsida, Silotum, Lycopsida, Selaginella and Lycopodium, Phenopsida, Equisetum, and Pteropsida, Dryopteris, Teres, and Adiantum. 3.4 Gymnosperm. The gymnosperm, gymnos, naked, sperma, seed, are plants in which the ovules are not enclosed by any aureole and remain exposed. both before and after fertilization. The seeds that develop post fertilization are not covered, that is, they are naked. Gymnosperms include medium-sized tree or tall trees and shrubs. Figure 3.4. One of the gymnosperm, the giant redwood tree sequoia, is one of the tallest tree species. The roots of generally taproots. Roots in the some genera have fungal association to form a mycorrhiza, pinus, while in some other cycas, small specialized root called coralloid root are associated with nitrogen-fixing cyanobacteria. The stems are unbranched in cycas or branched in pinus and cedrus. The leaves may be simple or compound. In cycas, the pinnate leaves persist for a few years. The leaves in gymnosperm are well adapted to withstand extreme of temperature, humidity and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperm are heterosporous. They produce haploid macrospore and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophyll, which are arranged spirally along an axis to form a lax or compact strobili or cones. The strobili bearing microsporophyll and microsporangia are considered or called microsporangiate or male strobili. Sorry. The microspores develop into the male gametophytic generation, which is highly reduced and is confined to only a little or limited number of cells. This reduced gametophyte is considered or called as a pollen grain. The development of pollen grains takes place within the microsporangia. The cone-bearing megasporophyll or with ovule or megasporangia are called the megasporangiate or female strobili. The male and female cone or strobili may be borne on same tree as in Pinus. However, in Cycas, male cones and megasporophyll are borne on different trees. The megaspore mother cell is differentiated from one of the cells of nucellus. The nucellus is protected by envelope and the composite structure is called an ovule. The ovule are borne on megasporophyll which may be clustered to form a female cone. The megaspore mother cell divides meiotically to form four megaspores. One of the megaspore enclosed within the megasporangium develop into a multicellular female gametophyte that bears two or more archegonia or female sex organ. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophyte and pteridophyte, in gymnosperm, the male and female gametophytes do not have an independent free-living existence. They remain within the sporangia retained on the sporophyte. The pollen grain is released from the microsporangium. They are carried in air current and come in contact with the opening of the ovule borne on megasporophyll. The pollen tube carrying the male gamete grows towards archegonia in the ovule and discharge their content near the mouth of archegonia. Following fertilization, zygote develop into an embryo and the ovule into seed. These seeds are not covered. Angiosperms. Unlike the gymnosperm, where the ovules are naked, in the angiosperms of flowering plants, the pollen grains and ovules are developed in the specialized structure called flower. In angiosperm, the seeds are enclosed in fruits. In angiosperm, the angiosperm are an exceptionally large group of plants occurring in wide range of habitats. They range in size from the smallest ulfia to tall tree of eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicines, and several other commercially important products. They are divided into two classes, the dicot and the monocotyledons. Figure 3.5. Summary. Plant kingdom include algae, bryophyte, pteridophyte, gymnosperm, and angiosperms. Algae are chlorophyll-bearing, simple thalloid, heterotrophic, and largely aquatic organisms. Depending on the type of pigment possessed and the type of food stored, algae are classified into three classes, namely chlorophyceae, phaeophyceae, and rhodophyceae. Algae usually reproduce vegetatively by fragmentation, asexually by formation of different types of spores, and sexually by formation of gametes, which may show isogamy, anisogamy, and oogamy. Bryophytes. Bryophytes are plants that can live in soil but are dependent on water for sexual reproduction. Their plant body is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by rhizoid. They possess root-like, leaf-like, and stem-like structure. The bryophytes is divided into liverworts and mosses. The plant body of liverwort is thalloid and dorsiventrally, whereas mosses have upright cylinder axis bearing spirally arranged leaves. The main plant body of bryophyte is gamete producing and is called a gametophyte. It bears the male sex organ called antheridia and the female sex organ called archegonia. The male and female gamete fuse to form a zygote which produce a multicellular body called the sporophyte. It produces haploid spore. The spore germinate to form gametophytes. In pteridophyte, the main plant body is a sporophyte which is differentiated into true root, stem and leaves. These organs possess well differentiated vascular tissue. The sporophyte bears sporangia which produce spores. The spores germinate to form gametophytes which require cool damp place to grow. The gametophytes bear male and female sex organ called antheridia and archegonia respectively. Water is required for transfer of male gametes to archegonium where zygote is formed. After fertilization, the zygote produces a sporophyte. The gymnosperm are the plants in which ovules are not enclosed by any ovary wall. After fertilization, the seed remain exposed and therefore these plants are called naked seeded plants. The gymnosperm produce microspores and megaspores which are produced in microsporangia and megasporangia, borne on sporophyll. The sporophylls, microsporophyll and megasporophyll are arranged spirally on axis to form male and female cone respectively. The pollen grain germinate and pollen tube release the male gamete into the ovule where it fuses with the egg cell in archegonium. Following fertilization, the zygote develops into embryo and the ovule into seed. The angiosperm are divided into two classes, the dicotyledons and the monocotyledons. Thank you. Thank you everyone.